Hi, hello everyone. Welcome back to my YouTube channel Sporifii Learn. Now let's see the module 5. I hope I have done like you have seen the module 1, 2, 3, 4. All the videos is uploaded. Please do check that. I have explained everything within 20 minutes or 15 minutes itself. Please do watch all videos. At last you can watch this module video. And tomorrow I'll be uploading like one short video for how to cover all the syllabus in one day itself, that I'll be uploading tomorrow. And if you're watching my channel without subscribing, make sure to subscribe and then watch the video. Now let's start the video without further delay. And please do hype the video also, which will be beside the comment box. And if you just press the hype button, it's sufficient. Please do hype the video also without forgetting. Okay, now let's start the first question, that is about the explain the use of electrical tongue. Electrical tongue and electronic nose in food science. Compare the e-tongue and e-nose with their biological counterparts. First, let's see what is electrical tongue. It's nothing but it is device used in food science to analyze the taste and flavor of food and beverages. Okay, electrical tongue is nothing but it is one device that is used for tasting of food and the flavor of the food and beverages. Okay, it measures the electrical properties such as conductivity, impedance, capacitance of a sample. Like it provides rapid, objective and non-invasive taste analysis without human taste testers. Without human taste testers, the device, if it is done, like it will only analyze and say what is the taste, and it will give the review at last. That is the basic idea of this electrical tongue. How the human tongue working is, see, first it is nothing but contains the taste buds, taste receptor cells. It is based on 5: sweet, salty, sour, bitter, umami, and it will be divided into saliva and enter the taste pores. That will be bind into the specific receptors on taste receptor cells. That binding generates electrical signals, and signals are transmitted to the brain through cranial nerves. Okay, the brain processes these signals and produces taste perception. Okay, now electrical tongue, how it works is, it will be having a sensor array in contact with the food sample. Okay, that will detect chemical and electrical properties related to the taste. Okay, the sensor responses are converted into the electrical signals. Now the sensor responses which we have got, it will be converted into the electrical signal. The signals are processed using pattern recognition and data analysis techniques. Okay, the system provides a numerical representation of taste and flavor characteristics it will be providing. This is the 5 working steps that the electrical tongue will be happening. I hope you have understood. See how what are the sensor used as. First is potentiometric ion selective electrodes. Next is voltammetric sensors. Next is impedance sensors. Next is optical sensors, conductometric sensors, and mass sensitive sensors. Why this potentiometric is used? Measure the concentration of specific ion. Next is voltammetric sensor is nothing but measure the current produced by oxidation or reduction reaction. Next is impedance. I hope you know that is represented by Z. That is measures the changes in electrical impedance. It is used to detect the sweet compounds. Okay, this is for bitter. This is for salty. Okay, next optical is nothing but for absorbance or fluorescence, that is for sour taste detection. Conductometric, it's nothing but for electrical conductivity, that designs the used for umami taste detection. That is another one type, right? Next is for mass sensitive, it detect the absorption by the taste compounds. It will be containing the taste, right? That the absorption it will do, and it will use the that. Okay, next is material uses. Polymers will be used, that is PVA, that is polyvinyl alcohol. I hope you know PVC will be saying, right? Next is polyethylene oxide, and next is metal oxide is tin dioxide and zinc oxide, SNO2 and ZNO, carbon nanotubes, that is high electrical conductivity and sensitivity. And next is dendrimers. Okay, functionalized nanostructures for specific taste detection it will be used. Microfluid devices made from silicon, glass or polymers it is used. Handle small volumes of samples efficiently it will be using. First, let's see human tongue. It have taste buds. This have sensors. This can be detect sweet, salt and all. This will be programmed for various tests. For this it will be one model. For this another one model. For this another one model. Like that it will be working for taste perception. And next for sensitivity, it is sensitive to low concentration. It is highly sensitive. Next is subjective and varies among individuals. Like one may have varies, like taste varies. Objective and it is standardized. Limitations is influenced by smell, temperature, texture. This cannot be fully mimic Human taste perception, this is slow process, this is fast. No calibration, requires calibration. Sensory evaluation, this is quick control and quality control and flavor profiling will be there. You can see the advantages. Non-invasive, does not require human testers. High throughput, provides objective, cost effective and quality. Limitations is cannot fully replicate human sensory experience what it will be having. Results can be affected by temperature, humidity and storage conditions and all, and it is like limited for understanding all the taste interactions which it has. And next is requires frequent calibration, may not capture the complete complexity of the food flavor perception. Next is bioengineering solution for osteoporosis, that is nothing but tissue engineering and bone regeneration. It is used for biodegradable scaffolds. 3D porous scaffolds are made from biomaterials, that is such as hydroxyapatite, tricalcium phosphate, collagen and biodegradable polymers. Next is stem cell therapy and also for growth factory, that is delivery. It will be used for such as bone morphogenic proteins and all, and VGF and all it will be using. Next, if we see the drug delivery system for targeted delivery and the controlled release, we will be using this. Next, if we see for 3D bioprinting, for patient specific bone grafts and bio inks will be used. Okay, that will be containing stem cell, collagen, gelatin, hydroxyapatite for growth and like for which is printed layer by layer and all for constructs. Okay, next is about the smart biomaterials, that is responsive materials. They can respond to pH, temperature or mechanical stress and release therapeutic agents when needed. Next is bioactive coatings. Next is mechanical stimulation technologies, that is low intensity vibration and electrical and electromagnetic simulation also it will be used, stimulation. Next is gene and molecular therapies. For gene therapy and RNA based approaches also it will be used. If we see the advantages, bone regeneration will be increasing, reduces the fracture risk. Next, it allows the targeted therapy with fewer side effects, enables the personalized treatment and may improve healing in severe bone defects. Okay. Next is limitation, high cost, long-term safety and effectiveness. Next is regularity and ethical challenges for stem cell and gene therapies. And next is the risk of immune reactions or implant failure may occur. These are the limitations in osteoporosis. Okay. Next is about the most commonly used bioprinting techniques and the materials in 3D bioprinting. Materials is hydrogels, cell-laden aggregates and decellularized extracellular matrix that is used, that is called as DECA. And next is synthetic polymer based bioinks, composite bioinks. Most commonly used bioprinting techniques is inkjet based, that is only majorly used, that is inkjet. Extrusion based bioprinting and nothing but next is laser assisted bioprinting. Next is microvalve based bioprinting, solid free form fabrication SFF. And steps, if we see, first the preparation of bioink will be done, design of the tissue structure will be done, printing, incubation and the assessment. For cell viability, tissue structure and the tissue functionality will be done. Next, if we see the advantages and disadvantages of bioimaging. Okay, advantages is improved accuracy, early detection, multimodal imaging it will be using, cost effective like less cost, and minimally invasive and or non-invasive itself, improved patient outcomes will be there, and advancement in medical research may occur. What are the limitations is high equipment cost will be high, exposure to radiation will be there, limitation like limited resolution in certain techniques. Next is requirement of skilled personnel and possibility of false results will be there, motion artifacts will be there, like contract agent risk, like for the contract agent risk will be occurring. And next is limited functional information will be there. Next, the one of the important question is like role of AI in disease diagnosis. This is one of the 100% question, don't skip this. Okay, AI is transforming disease diagnosis by providing accurate, efficient and data driven healthcare solutions. Okay, it will first analyze medical data and recommend the treatment also, and it will give accuracy and supports the clinical decision making. What is the application? See, first image analysis, x-rays, CT scans, MRI scans and all can be done, like known as CAD. Okay, computer aided diagnosis can be done. Example, detecting cancerous tumors from like tumors can be detected from MRI images. Next is in data analysis. AI analysis large volume of patient data, uses electronic health records, laboratory reports and medical histories. Next is identifies disease patterns and trends. Next is example is like risk of diabetes or what is the rate that the person have for heart disease. Next is disease diagnosis. AI evaluates the symptom, test results and patient information, and it will give accurate diagnosis. Next is for personalized medicine for a single person. What are the based on the input they have given for age factor or the like what are the input given, based on that individual it will be giving clinical decision support. Also it will be having like tests, choose effective and manage the patient care effectively. Advantages, if you see, that is improved diagnostic accuracy will be there, faster diagnosis, early detection of disease, personalized treatment, efficient data handling, clinical decision support. If you see the limitation here, that is lack of understanding of algorithms, okay, bias in AI model, like something it may fail, okay, regulatory challenges, high cost and dependence on data quality, privacy and security reasons also may occur one time. Next is about the DNA origami and biocomputing. DNA origami is nothing but one nanotechnology technique in DNA molecules. They are folded into two specific like two-dimensional or three-dimensional shapes. Okay, they are the long strand is called as scaffold, which we studied in previously, and that is when it shortly folded, and complementary DNA strands are called as staples. Okay, next is it is widely used in nanotechnology, biomedicine and materials science. Technology's importance is nanoscale manufacturing, drug delivery, biosensors, biomedical imaging, gene therapy, biocatalysis and nano patterning it is used. I am not going deeper because you have to study, right? Like you can see the headings and you can write up the theory, so I am not going into much deeper. Advantages, if you see, programmability, versatility, high precision will be there, functionality and biocompatibility, like it is naturally occurring and less likely to trigger immune responses. Next is about the limitations is complexity, high cost and stability issues and scalability challenges. Next, biocomputing, it's nothing but the use of biological systems such as DNA, enzymes and living cells for computing and information processing. It combines biology, computer science and engineering to perform the computational tasks. Okay. First is technological information, like importance, if we see, computational power, data storage, medical application, environmental monitoring, energy efficiency, robustness and the versatility. If we see the advantages, biocompatibility is energy efficiency, scalability, robustness and flexibility. If we see the limitations, that is having low speed, like slow it will be working a bit, and complexity and reliability issues will be there, and the high cost. Okay, these are the limitations of biocomputing. I hope you have understood. Next we'll be seeing about the removal of heavy metals by biomining, okay, and bioremediation through microbial surface adsorption. Okay, bioremediation, it's nothing but use of microorganism, plants or animals to remove the contaminants from soil, water or air. This is nothing but using the ores, okay, ore deposits, okay, that is such as lead, cadmium, mercury, arsenic. Okay, arsenic, cadmium, lead and mercury it will be using, and comparison also have given. See here, it is nothing but remove from environment. This is from like extract valuable metal from ore. Adsorb and degrade, this is extract from metals, and this is organic pollutants and contaminants, it is the target. Valuable metals and minerals, this is pollutant degrading microbes, this is metal adsorbing. Restores ecosystem quality, this is for the mining disturbances. Next is months to years it may take, this is comparatively faster. Soil, water, air remediation can be done. This is metal extraction and mining can be done. If we see process, that is first it will be identifying the contaminant site, and next it will be isolating and characterization of the metal resistant microbes will be done. Next the culturing and enrichment of the collected will be done. Next is preparation of microbial suspension. Next is application to the contaminated site, and next is about the microbial absorption and sequestration, and next is separation and removal of metals. Metal resistant microorganisms is nothing but lead, cadmium, mercury, arsenic, as we studied. You can go through this. Methods used is Phytoremediation, chemical extraction, biosorption, physical removal also can be done. Electrochemical methods can be used. Okay, advantages is eco-friendly, environmental friendly, cost effective, selective, effective, and sustainable. If we see the limitations, slow process, incomplete removal can be there in microbial like biomining, and next is microbial inhibition, and next is difficulty in harvesting, limited application. Next, if we see the question, how the muscular system and skeletal system acts as natural scaffolds. I have said about scaffolds, so I hope you can see this question. See, it is structural support, attachment sites for muscles will be there, protection of organs it will help in, support for tissue growth and mineral storage it will be helping. That is, skeletal system acts as a natural scaffold, but muscular system it supports and like it will give the support and stability to the body, framework for soft tissues, and next is force generation and movement, tissue repair and regeneration, and mechanical support it will be giving. Next, the last question is about the write a short note on self-healing concrete. Okay, the self-healing bioconcrete is a special type of concrete that can repair on its own cracks. Okay, that is it contains the bacillus bacteria and calcium lactate as a nutrient source. Will be using as bacillus bacteria and calcium lactate. When cracks develop, the bacteria become active and produce calcium carbonate, that is CaCO3, will fills the and seals the crack. This process is called as biomineralization. That is, you have to remember this. First, see, the bacillus bacteria will be there and calcium lactate and concrete matrix. How this will be working? We'll be seeing. First, bacillus bacteria and calcium lactate are mixed with concrete. Then the bacteria remain dormant and inside the concrete. Then the crack will develop in the concrete and water and oxygen enters the cracks, and bacteria will become activated. Activated bacteria produce calcium carbonate, that is CaCO3. Calcium carbonate fills and seals the cracks. Okay, it will be filling and sealing the cracks. Structural integrity of the concrete is restored. Next is working principle is when crack forms, moisture and organized oxygen will be there. It will be entering the concrete. These conditions activate the dormant bacillus bacteria. The bacteria consume calcium lactate and produce calcium carbonate. It will be producing. The produced calcium carbonate, it will be acting as a natural cementing material. The cracks are filled and repaired automatically. Next is, if you see technological importance, like increased durability, sustainability, reduced cost, increased longevity, that is it will be for lifespan is more. And next is new engineering applications also, such as bridges, tunnels, dams and all difficult to maintain structures and all, it will be used, and reduce carbon footprint. If we see the advantages, that is self-repairing capability, increased durability and strength, and reduced maintenance and repair cost, environment friendly, improved resistance to water penetration, longer service of structures. If we see the limitations, higher cost, initial cost, technology is still under research and development. Limited effectiveness for large, like very large cracks, if it is there, it is limited effectiveness. Requires suitable environmental condition for the bacterial activity. Large scale implementation is still challenging. Like still, I said right, like research and development is happening. So it is major more time. That is about the bioconcrete. I hope you have understood all the 9 questions which I have explained, and that's all I wanted to say in this today's video. I hope you have understood everything. And that's all. And tomorrow I'll be uploading one short video also that will be containing all the 5 modules in one video. I'll be trying to complete it within 1 hour itself. And if you have liked this video, please make sure to like, share, comment and subscribe to my YouTube channel. Thank you for watching until last. Meet you in the next video. And until next video, bye bye.